we come to the final chapter of this course that deals with metabolism. Okay. Now, in metabolism, we need certain nutrients. We are going to learn how the food that we take in, especially in the metabolism of carbohydrates, the food that we taking, how that is broken down. Okay. Now, metabolism actually, as you have learned from your school days, comprised of catabolism, which is the degradation of nutrients to generate energy and starting materials, which is what we are going to do basically. And anabolism is the biosynthesis of biomolecules from starting materials, which also actually happens in our body because we create proteins. Proteins are synthesized in the body, which is a process that would require anabolism. Catabolism is the breaking down of the nutrients that is ultimately going to provide the energy for the actions or whatever work that we do. The metabolites that take part in these processes actually are substrates and intermediates, and of course, there will be a large number of enzymes that are going to be actually part of the whole system. Okay. Now, if we just look at this whole picture, and I will show you another one also where we will see how the whole process actually takes place. We have energy containing nutrients that we take in our diet: carbohydrates, fats and proteins. In the catabolism, that is in the breakdown of these processes or the breakdown of these units, we actually have energy depleted end products that are finally carbon dioxide, water and ammonia. Okay. And we have in this process then the use or the utilization of certain aspects or certain cofactors that we have already looked at. We have ADP and ATP in a large extent, we have NAD plus, we have FAD, we have NADP plus and so on and so forth. Now, all of these actually will then either break down or form in different ways, giving us finally the chemical energy. Then we have certain precursor molecules that are also found in our body, where we have the amino acids, the sugars, the fatty acids and the nitrogenous bases that in a process of anabolism, that is in the formation of the macromolecules, will ultimately lead to the cell macromolecules such as proteins, polysaccharides, lipids and nucleic acids. Okay. So, eventually it is just like a whole cyclic process that it is the food that we take that is ultimately broken down, and then after the breaking down, the bits and pieces, some of them get lost in energy depleted products, some of them form the precursor molecules for the formation or the generation of other proteins, polysaccharides and lipids and nucleic acids. Now, if we look at actually the whole process, it is extremely interesting. Okay. This is like a system of a gear system. Okay. We have the process of glycolysis, which we are going to study in our breakdown of glucose, okay, in the metabolism that we are studying. The glycolysis leads to something called the TCA cycle, which is the tricarboxylic acid cycle. That has electron transport. The electron transport we just looked at has a proton pump to it. The proton pump then uses ATP synthase to produce ATP, that is cellular work, where we get work. This oxygen again comes in from the blood, utilized in the ATP synthase in the electron transport system, where we have the reduction of the oxygen to water. Now, this is the way all of these are interconnected actually, and if you just look at a whole picture of all the metabolic pathways, there is actually a beautiful picture on the net that has all the metabolic pathways that actually take place in our body. What I am going to give you is just probably a small like drop in the huge ocean of all those metabolic pathways that actually take place. Okay. Now, what we actually intake, we are looking at stages of catabolism. Our intake is proteins, polysaccharides, lipids, that is fats, proteins and carbohydrates basically, that is what we intake. The smaller blocks of proteins break down, what is the breakdown of proteins going to give you? Amino acids. The breakdown of polysaccharides is going to give you monosaccharides. The breakdown of lipids is going to give you fatty acids or glycerol. Fine. Now, each of these again are further going to be broken down and converge to acetyl coenzyme A, which is an extremely important part in the catabolic pathway that is going to take you again to the citric acid cycle that requires the oxidation of acetyl coenzyme A. So, basically what we are getting at is we are going to look at just this part here, monosaccharides, the breakdown of glucose, that is what we are going to look at. But if you consider the umpteen polysaccharides that are present in the body that are possible for breaking down, you realize that each of them is an enzymatic reaction that is required for the breaking down from its polymeric form to the monomeric form, which is finally going to be broken down into other forms. So, what we have is we have our lipids, polysaccharides and proteins, lipids breaking down into fatty acids that form the acetyl CoA, polysaccharides breaking down into glucose, the monomeric unit, in the process of glycolysis forming pyruvate, which is what we will see in the process of glycolysis that we will study. Proteins again breaking down into amino acids that again are utilized in this acetyl CoA, which finally is used in the tricarboxylic acid cycle, which is going to be the breakdown of the tricarboxylic acids, the oxaloacetate and so on and so forth. Then we have this process where we have FADH2 plus NADH giving you with oxygen oxidative phosphorylation in the production of ATP. Okay? This is a part that we have studied in a bit detail, and now what we are going to look at is we are going to look at the glucose going to pyruvate in the process called glycolysis, and in the event producing ATP also, but requiring ATP also in a number of steps, and finally acetyl CoA that is going to be utilized in the tricarboxylic acid cycle that is also known as the Krebs cycle. Okay. Now, before I get into the process of glycolysis, this is something that we looked at before when we considered the vitamins. I mentioned that each of these vitamins, you now realize, is the precursor for a large number of cofactors and prosthetic groups. Okay? Coenzyme A is another such compound that is extremely important in the formation or the transfer rather of the acetyl group, that is a transfer of a 2 carbon system for any of the processes that occur. What we have in coenzyme A is actually an ADP part, an adenine dinucleotide part, a phosphorylated one because there is a phosphor at this position if you can see, and we have pantothenic acid. Okay, that is one vitamin, and to it we have mercaptoethanolamine attached. Now, this SH, if acetylated, is called acetyl CoA. Okay? So, whenever we look at any metabolic pathway, you will see acetyl CoA come into the picture a large number of times. Okay? But you have to remember it is derived from that vitamin, and it is nothing but a phosphorylated ADP linked with an oxygen to pantothenic acid that is linked to mercaptoethylamine. This unit is coenzyme A. If this thiol group is acetylated, it becomes acetyl CoA, and that is exactly as to how it is referred to in all of the metabolic pathways, acetyl CoA. Okay? So, it is this acetyl part that is important, and it is this acetyl part that is important, and the rest you realize is derived from phosphorylated ADP, vitamin pantothenic acid and mercaptoethylamine. Now, if we look at all the catabolic processes that occur, the glycolysis pathway will lead us to pyruvate. Pyruvate, all of the breakdown getting to acetyl CoA being the main part of the TCA cycle that ultimately breaks it down to carbon dioxide and water. Okay? Now, this is obviously going to involve a large number of enzymes, okay? And we will look at each of those enzymes and how they act. Okay. Now, glycolysis. The process of glycolysis takes place in the cytosol of cells, and glucose enters the glycolysis pathway by the formation of glucose 6 phosphate. Now, glucose 6 phosphate means that we are going to have a large number of steps that are actually going to get you to pyruvate. There are 10 steps involved, which means there are 10 enzymes involved, and we have this form pyruvate, and we have 3 steps that are regulated in the formation of pyruvate from glucose in the 10 steps that form the process of glycolysis. Okay? So, we have glucose go to pyruvate in 10 steps, and we have 3 of those steps that are regulated, and it is occurring in the cytosol of the cells. If we do not have oxygen, then this pyruvate forms lactate. Okay? This is the overall reaction. We have glucose plus 2 ADP plus 2 PI, that is phosphate, plus 2 NAD plus go to 2 pyruvate plus 2 ATP. So, you are generating ATP here also, plus 2 NADH plus H2O. Okay? So, we are going to look at each of these steps and see how you have ATP consumption, ATP production, and finally, we are going to take an account of where we are using ATP, where we are producing ATP. Okay? Now, before we get into that, we have to see what enzymes are involved in the pathway, in the metabolic pathway. We have kinases, isomerases, aldolases, dehydrogenases. Okay? Kinases are a specific class of transferases, and kinases are those that transfer a phosphate group. This we have looked at before, or I have mentioned before, where we can have, we will see how hexokinase will be a part of our glycolysis mechanism. So, we have kinases that transfer a phosphate group from ATP to a specific substrate. We have isomerases. What are isomerases going to do? They are going to convert one isomer to another. That is what the function of isomerases is. If we look at aldolases, they are going to catalyze aldol condensations. All of you know what aldol condensations are. So, we have kinases, isomerases, aldolases. We have dehydrogenases. Dehydrogenases we have looked at also, where we have the removal of hydrogens by oxidation. Okay. Then what are the other enzymes? We have a few more. We have mutases that are actually group transfer enzymes. They transfer phosphate from one position of the substrate to another position. Okay. So, say you have a glucose 1 phosphate. To form a glucose 6 phosphate, you will need a mutase. Okay. So, the mutase is going to be a group transfer enzyme that is going to transfer the, say in this, the common use would be the transfer of phosphate to a different position. An example being glucose 1P mutase, which would actually transfer the phosphate from the carbon 1 to carbon 6. Okay. Forming from glucose 1 phosphate, it would form glucose 6 phosphate. In that case, you would need mutase. If you want an enolase, you would convert a C double bond C group to an alcohol. Okay? Because you have to remember that when we are looking at each of these steps, we are finally going to break down glucose into carbon dioxide and water. That is our final aim. Okay? So, we have to look at how this can be accomplished with this in the biological way in certain with the use of these certain enzymes. We have synthase, that is also known as synthetase. What does that do? Just combines two molecules together, synthesis. We have ATPases that are going to hydrolyze ATP to ADP and PI, and this is in reverse to ATP synthase, which was, what was ATP synthase doing? It was taking ADP and PI and producing ATP. This hydrolyzes ATP to ADP and PI. Okay? So, these are the pathway enzymes that are going to be used in every step of the way as we go along. The first step, that's step number 1, is the use of hexokinase. Hexokinase, so the first step in glycolysis after you've broken down the polysaccharide to glucose, okay, we now have glucose. We're not going into how the polysaccharide is broken down or how even the other dietary nutrients are broken down like fats or amino acids, proteins or whatever. We're just going to look at the metabolism of carbohydrates, just looking at the breakdown of glucose. The glucose breakdown will ultimately lead us to pyruvate and it will involve 10 steps. This is step number 1, where from glucose we form glucose 6-phosphate. That means the 6th carbon atom is no longer OH, it is now phosphorylated. It is phosphorylated by taking a phosphate from ATP. In the event, ATP becomes ADP. Okay? This is a coupled reaction. We will look at the energetics of these steps. The glucose going to glucose 6-phosphate has a delta G that is positive. The ATP to ADP has a delta G that is negative. A coupled reaction will give a favorable forward reaction to this, and the reaction actually involves a nucleophilic attack of the hydroxyl OH that is attached to carbon 6 to the gamma phosphate of ATP, resulting in the formation of ADP. And in this case, the enzyme that is involved is hexokinase. Hexo means it is going to act on a 6-membered sugar ring. It is a kinase because it helps in the transfer of the phosphate, and the ATP binds to the enzyme as a complex with magnesium. Okay. Now, why would we have magnesium there? What is ATP? ATP has a large number of negative groups. Right? There are 3 phosphates one after the other. What magnesium does, it helps in the it interacts with the negatively charged phosphate oxygen atoms and provides charge compensation, and also promotes a favorable conformation of ATP at the active site of the hexokinase enzyme. Because you have to realize that when we're looking, we're not going to look at the details of how the enzyme is working, but what we have here is we have magnesium in this set so that the ATP can be favorably interacted with the magnesium, because we are finally going to break the final phosphate bond here. And what is going to happen? Where is the phosphate going to go? It is going to be attached to the 6th carbon atom of glucose in the formation of glucose 6-phosphate from glucose. Okay. So, that is our step number 1. So, step number 1 is glucose to glucose 6-phosphate. The enzyme is hexokinase, and you have ATP going to ADP and PI, and not PI, PI is attached to the 6th carbon. So, ADP. Okay. Now, this reaction is highly spontaneous because the phosphoanhydride bond of ATP is cleaved. Okay. That is a high energy bond as we call it, and the phosphate ester has a lower delta G of hydrolysis. Now, what happens in this case is you have what is called an induced fit. Remember, we studied induced fit for the enzyme mechanisms, the way they work. There is a lock and key mechanism and an induced fit mechanism. Usually, the enzymes of the glycolytic pathway have induced fit. They are not ones that would just sit with a lock and key where an exact substrate would come and sit there. It happens such that the binding of the glucose to this hexokinase promotes a conformational change in the protein, in the enzyme rather, and it stabilizes a different conformation whereby the ATP is positioned in such a way that the terminal phosphate can be transferred to the substrate, that is glucose. Okay. So, what happens is you normally would have hexokinase. As soon as the substrate glucose comes into the picture, there is the ATP that is positioned favorably so that the phosphate can be transferred from where? From ATP to glucose. Okay. So, and we also have this important point where water is excluded from the active site, because what would happen if water would be there? ATP would be hydrolyzed. Okay. We do not want the hydrolysis of ATP, we want the phosphate to be transferred only to glucose. Okay. So, once the substrate comes to hexokinase, it changes its conformation so that it can accommodate glucose, at the same time positioning ATP in such a manner so that it is favorably interacting. And remember what happened, how did the reaction take place? It was the 6th OH, the OH attached to the 6th carbon atom that actually went and attacked the phosphate bond here. This is step number 2. In step number 2, you have glucose 6 phosphate go to fructose 6 phosphate. So, what happens is you have the formation of an isomer. So, the enzyme involved is an isomerase. Okay. What is this isomerase? It is a phosphoglucose isomerase, and what is happening here is the glucose 6 phosphate forms a fructose 6 phosphate, where instead of the aldehyde now, the aldose, you have a ketose, right? You have the CH2OH up here now. Okay. So, we have glucose 6 phosphate go to fructose 6 phosphate. The enzyme involved is phosphoglucose isomerase, and the mechanism actually involves acid base catalysis with ring opening via an diolate intermediate, and finally, we have ring closure. So, we have an isomerase. So, what was the first step? First step was glucose to glucose 6 phosphate. The next step is glucose 6 phosphate to fructose 6 phosphate. What are the enzymes required? In the first step, we need a kinase. In the second step, we need an isomerase. Okay. Third step. Look at what is happening. We had fructose 6 phosphate go to fructose 1,6 bisphosphate. So, what is happening? I have the addition of another phosphate at the 1 position. So, what is the enzyme that I need? A kinase. That is what you have to identify. You have a process, you know the substrate, you know the product. What is the enzyme involved? You know in this case that you have prepared fructose 6 phosphate from where? From glucose 6 phosphate. And it was just an isomerization, so you needed an isomerase. You are going now from fructose 6 phosphate to fructose 1,6 bisphosphate, which means you have added another phosphate, which means the breakdown of another ATP, which means that you need a kinase. And what kind of a kinase do you need? A fructokinase. Why a fructokinase? Because you have a fructose and you are adding a phosphate to a fructose. That's it. Okay. So, you have fructose 6 phosphate that is going to from fructose 1,6 bisphosphate, and this process is actually highly spontaneous, and this phosphofructokinase reaction is the rate limiting step of glycolysis. We will see how that is later on when we studied the whole process, and this enzyme is extremely tightly regulated. There are some processes, when we go through the whole system, you will see there are some processes in the glycolysis steps of reactions that are reversible. There are some that are irreversible. Okay? Means you cannot, once glucose has got into glucose 6 phosphate, there is no way it is going to get back to glucose. Okay? The next step. What we have here now is we have fructose 1,6-bisphosphate. Now, what happens to fructose 1,6-bisphosphate? If you look at the previous, we have it in the ring formation here. Okay. In the next step, there is going to be the breakdown of this. How many carbons do we have here? 6. Glucose has 6 carbons. We have 6 carbons here. We have 1, 2, 3, 4, 5, 6. What is going to happen now is, in the next step, first there is ring opening. Then the 6-membered ring is going to break down into 2 3-membered rings. Okay? Because you have to remember that finally we have to get to carbon dioxide and water. Okay? So, unless we start chopping up, it is not going to be possible. So, we have our glucose. So, now we are going to go to a step where we are going to break the fructose 1,6-bisphosphate into 2 3-carbon units. The process here is a reverse of aldol condensation. It is aldol cleavage. We have here the fructose 1,6-bisphosphate, okay, which is now not in its ring form, but in its open form, where you can see the ketone. It is a ketose, right? So, it has to have this C double bond O. There are 2 phosphates attached to it now, one at the 1 position and one at the 6 position. Fine. We have now an aldolase, which is a reaction that is going to involve aldol cleavage, and there is going to be a reverse of aldol condensation, which means you are going to have a break at this position here, where we are going to have the formation of 2 3-carbon units. One of them is dihydroxyacetone phosphate. If you look at this, this is acetone, CH3, CO, CH3 is acetone, right? This is acetone, CH3, this is acetone. Dihydroxyacetone, is that, right? Now, if we have dihydroxyacetone phosphate, we have phosphorylated one of these. Okay? So, that is exactly what we have. Here we have dihydroxyacetone phosphate and we have glyceraldehyde 3-phosphate. This is glyceraldehyde. You recognize the CHO of the aldehyde, the CHOH and CH2OH which would have been here, but we now have it phosphorylated. So, the phosphor now is distributed in the 2 3-carbon units, right. So, this is the ketone form, this is the aldehyde form. What are these? Isomers. So, you can go from one to the other by what enzyme? An isomerase. What is this isomerase going to be named? It works on a 3-carbon unit. So, a triose phosphate isomerase. Okay? So, you see, if you look at the nomenclature, it is actually very simple. All you have to know is what the substrates are and what the products are. In this case, we have an aldolase with the reaction being an aldol cleavage. We have the formation of dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So, this step is where you have the breakdown of the 6-membered or the 6-carbon unit. Okay? Now, if we look at how this 6 carbon unit or the aldolase actually works, there is a lysine residue. Where is this lysine residue? In the enzyme aldolase. Obviously, it has to be there because the aldolase is what is acting on the substrate. What is the substrate? The substrate is fructose 1,6-bisphosphate. That is your substrate. What are your products? Your products are dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Okay, so you have glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which later on you will see it's written as DHAP and G3P. Okay, so we have a lysine residue in aldolase that is present at the active site. What this lysine does is the keto group of fructose 1,6-bisphosphate reacts with the amino group of the lysine, okay, and it forms a protonated Schiff base. Then there is a cleavage between carbon atoms 3 and 4. We are not going into go into the details of all this, but what you have to know is you have the breakdown from the 6 carbon to the 3 carbon by the enzyme aldolase. The enzyme has a lysine residue that interacts the epsilon, the amino group of the lysine interacts. Where is this lysine? It's in the active site of aldolase. It interacts with the keto group of fructose 1,6-bisphosphate and it breaks it up. Okay, there is cleavage between carbon atoms 3 and 4. Okay, so this is just the triose phosphate isomerase that is going to be interconverting dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So, in this protein actually is pretty interesting in its structure also, but we are not going into the details of that. It's called TIM, where it has a TIM barrel actually associated with it. Okay, we have the glycolysis that is going to continue from glyceraldehyde 3-phosphate. Now, the equilibrium constant. Now, so what is the triose phosphate isomerase isomerizing? What is it doing? It is converting dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. So, this is an isomerization reaction. The equilibrium constant is such that it favors dihydroxyacetone phosphate. Okay? So, this is favored, but the glycolysis steps, the further steps actually continue from glyceraldehyde 3-phosphate. So, if the equilibrium shifts to dihydroxyacetone phosphate, what does it mean? It means I may not have sufficient glyceraldehyde 3-phosphate to continue with the glycolysis. I will repeat that once more. We have the enzyme triose phosphate isomerase. The equilibrium constant of triose phosphate isomerase is such that it favors dihydroxyacetone phosphate. Okay? When you have an equilibrium, your equilibrium either shifts to the left or the right, to the reactants or the products. In this case, dihydroxyacetone phosphate is preferred based on its equilibrium constant. But the process of glycolysis will only continue with glyceraldehyde 3-phosphate. So, what has to be done? Your equilibrium has to be shifted to the right. So, what is usually done is this glyceraldehyde 3-phosphate, as soon as it is formed, it is then utilized in the next step. So, if it is utilized in the next step, what happens? Your equilibrium is shifted to the right, which means some more of the dihydroxyacetone phosphate has to be isomerized to glyceraldehyde 3-phosphate. It's regulated. You see how it is regulated. Okay? The equilibrium is such that it's shifted to the left. But if you require the breakdown of the glyceraldehyde 3-phosphate, you break it down. What happens then? Your equilibrium is moved in such a way that the dihydroxyacetone phosphate forms glyceraldehyde 3-phosphate. Okay, is that clear? So then what happens? Then there is removal of the glyceraldehyde 3-phosphate and subsequent spontaneous reaction. Okay? But it does not happen as it is. The equilibrium shifted to the left, but with the removal of the product, that is glyceraldehyde 3-phosphate, it obviously moves to isomerize more of the DHAP. Okay, now this is how it actually, this is the intermediate that you have, which is the enediol intermediate, where you have the ketose aldose conversion, which actually involves an acid base catalysis, and the isomerase that actually takes place is phosphoglucose isomerase, or rather what happens here is, when we have dihydroxyacetone phosphate, what are we talking of? We are talking of a ketose, right? When we have the glyceraldehyde 3-phosphate, we are talking of an aldehyde, right? So there has to be an enediol intermediate that is going to take you from the ketose to the aldehyde. It's similar to fructose and glucose. What is glucose? Glucose is an aldose and fructose is a ketose. You have an isomerization there also, right? That takes you from glucose to fructose. It's exactly the same thing, but here you are working on a 3-carbon atom instead of a 6-carbon. That's the difference. Okay? So we have the dihydroxyacetone phosphate, we have the glyceraldehyde 3-phosphate, and these are formed from an enediol intermediate. So when we are talking of the triose phosphate isomerase, we are talking of this equilibrium, right? It is this equilibrium that we are talking about. So the equilibrium actually will shift to the left side, but with the removal of glyceraldehyde 3-phosphate, what will happen? There will be more formation of the glyceraldehyde 3-phosphate that will eventually be used in the other steps of glycolysis. Okay? So this is what we are going to do for glycolysis for today, because there are some other discussions that I want to make regarding hexokinase. Okay. Now we have glucose. So these are the steps we have done up till, we just broken down the 6-membered ring. Let's put it that way. We have broken down the 6-membered ring. We will see now what is going to happen to the 3-carbon ring. Okay. The 3-carbon, not the ring, the 3-carbon system. Now the 3-carbon system will eventually get into the tricarboxylic acid cycle, and that tricarboxylic acid cycle will ultimately produce carbon dioxide and water. So you will have it broken down. But what we started off with was, we started off with glucose. In the first step, we had glucose go to glucose 6-phosphate, right? Now since it took up a phosphate, it required the breakdown of an ATP. It's just summarizing the steps that we've done so far. So we have glucose to glucose 6-phosphate, that required the breakdown of an ATP, and the enzyme was hexokinase. Step number 1 in our glycolysis. Step number 2 was the formation of fructose 6-phosphate from glucose 6-phosphate, which is nothing but an isomerization, where we have an aldose go to a ketose. The enzyme is a phosphoglucose isomerase. Okay. Step number 3, where we have our fructose 6-phosphate form fructose 1,6-bis phosphate. What is happening there? I am adding another phosphate. In the addition of the phosphate, I have to break down another ATP, and I need another kinase, right? So I have fructose 6-phosphate that has taken up, will take up phosphate from the ATP into forming fructose 1,6-bis phosphate with the help of phosphofructokinase. The next step now is the breakdown of the 6-carbon to 2 3-carbons, and we have aldolase. Aldolase is going to break down fructose 1,6-bis phosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. And if you notice here, it is glyceraldehyde 3-phosphate that will allow the glycolysis to continue. So, we need an isomerase that is going to transform our dihydroxyacetone phosphate to glyceraldehyde 3-phosphate. Okay. Now, in the steps that we wrote here, if you notice, the steps that involve the addition of the phosphate are irreversible. Okay. So, glucose forms glucose 6-phosphate with the formation of ATP, the breakdown of ATP to ADP. That step is one way. Okay. That means once glucose enters the cell and forms glucose 6-phosphate, it is trapped into being broken down. Okay. If it is not required to be broken down, then glucose will actually go for storage as glycogen. Okay. But once it forms glucose 6-phosphate, it has to be broken down. Okay. So, if we look at the features actually of hexokinase, this enzyme is inhibited by its product. Okay. Now, biochemically that makes absolute sense, because if hexokinase is broken down by glucose 6-phosphate, is inhibited by glucose 6-phosphate, then what is it going to do? It is going to prevent the formation of glucose 6-phosphate from glucose. So, it is going to be regulatory in nature. So, as soon as there is sufficient glucose 6-phosphate or sufficient glucose to be broken down, it will stop itself from acting, because you do not want all the glucose to form glucose 6-phosphate, because once the glucose 6-phosphate is formed, it has to be broken down. It's entered the glycolytic cycle, there is no way it can go back. Okay? But if hexokinase is inhibited, then what happens? Glucose can still go someplace else and be stored as glycogen in the liver, say. But once hexokinase has acted on glucose, there is no way it can go back. Right? So, glucose 6-phosphate acts as an inhibitor to hexokinase, so that further glucose breakdown is not possible. Here, that is exactly what I was saying. Hexokinase is inhibited by its product glucose 6-phosphate, and the way it inhibits hexokinase is by competition at the active site as well as by allosteric interactions at a separate site on the enzyme. What does that mean? It means that it will change the active site conformation to such an extent that it will not be able to bind the substrate glucose. And remember, I showed you a rough cartoon of hexokinase where it acts, it has an induced fit as soon as glucose comes into the picture, right? And then the ATP is positioned in such a manner that it forms glucose 6-phosphate. Okay? But if glucose 6-phosphate actually inhibits hexokinase, then it will either inhibit by sitting at the active site itself, or it could inhibit at another position where it could affect the active site so that the substrate cannot bind. So, once the substrate cannot bind, it means that the enzyme is inhibited. If the enzyme is inhibited, what happens? Then the product will not be formed. If the product is not formed in this case, it means that glucose breakdown does not occur. As simple as that. Okay? So, it is extremely tightly regulated. So, the cells trap glucose by phosphorylating it. So, once glucose is phosphorylated, it is trapped. It has to be broken down, right? But this can be prevented if this glucose 6-phosphate inhibits the enzyme. If hexokinase is inhibited, then glucose cannot form glucose 6-phosphate. Fine. So, the product inhibition of hexokinase ensures that cells will not continue to accumulate glucose from the blood. Okay? If glucose 6-phosphate within the cell is in sufficient quantity. Okay? Because once your glucose 6 phosphate is in sufficient quantity, it means that this can continue in the glycolytic steps. But if you need it, then only glucose is broken down. Unnecessarily, glucose is not broken down in the cell. Okay? It is only when the glucocarbohydrate metabolism is required is glucose broken down in the cell, because once this first step of the glycolytic cycle takes place, it is irreversible. Okay? Now, glucokinase, which is a variant of hexokinase, is found in the liver. This has a high Km value. All of you know what the Km value is now, Michaelis-Menten constant for glucose, and is active only at high glucose concentrations. So, the glucokinase acts at high glucose concentrations. So, when there is a high level of glucose concentration, the glucokinase enzyme comes into the picture. The glucokinase enzyme is not subject to product inhibition by glucose 6 phosphate. What does that mean? It means that glucokinase will act on glucose. Because why? It is not inhibited by glucose 6 phosphate. So, any glucose that comes into contact with glucokinase will have a phosphate transfer to it. As simple as that. Why is that? Because the glucokinase is not subject to product inhibition by glucose 6 phosphate. So, glucokinase actually works at high glucose concentrations. So, when there is high glucose, glucokinase will transfer a phosphate to the glucose at high glucose concentrations. Now, liver will, that means where is this glucokinase found? It is found in the liver. What it does is it takes up and phosphorylates glucose even when the glucose 6 phosphate is high. Why is that? Because it is not inhibited by glucose 6 phosphate. Glucose 6 phosphate has nothing to do with glucokinase. If it was hexokinase, then glucose 6 phosphate would have inhibited the enzyme. But glucokinase is not inhibited by glucose 6 phosphate. So, it is immaterial whether glucose 6 phosphate is high or low. It does not matter. Liver glucokinase is actually inhibited by a glucokinase regulatory protein. What does that do? You have to remember that when we are considering an inhibition, it is to prevent the enzyme from forming its product. Okay? Now, naturally, there are certain steps that you would not want the product to be formed. For example, in the hexokinase set. In the hexokinase step, you realize that breaking down of all the glucose present is extremely unnatural. Okay? And you wouldn't obviously want that to happen because you want some stored glucose. Okay? So, the product inhibition in the case of hexokinase is an extremely clever way of preventing the glucose from being broken down. But in the case in the liver, the glucokinase is present, not hexokinase. It's a variant of hexokinase, but it acts only when the glucose level is very high. Okay? Now, the liver glucokinase is subject to inhibition by another protein, and the protein will regulate when glucokinase is going to act. Okay? So, it is not inhibited by a product, it is inhibited by another protein that is going to regulate the action of glucokinase as to when it should be acting on a glucose and when it should not be acting on a glucose. Okay? This glucokinase regulatory protein actually would work then when we have high glucose. Okay? Because that, I mean, when we have low glucose, because high glucose would actually have glucokinase work on it. So, what happens in this case, glucokinase with its high Km value for glucose allows the liver to store glucose as glycogen when the blood glucose level is high. Okay. So, when the blood glucose level concentration is high, it will allow the storage of glucose as glycogen. Now, what happens is we have, therefore, this is what we would have in the liver, where we would have the glucose acted upon by the Gluco Kinase to form glucose 6 phosphate. It would not be broken down, or rather Hexokinase would be inhibited by glucose 6 phosphate, but not Gluco Kinase. Okay? And then we would have from the glucose 6 phosphate, once this is formed in the cell, it is trapped. Okay? So, we have a regulatory mechanism that actually works here. Then glucose 6 phosphatase catalyzes the hydrolytic release of Pi from glucose. So, what happens if you look at the step here, you had glucose 6 phosphate formed, but there is another protein, that is glucose 6 phosphatase, that can release the phosphate, but this happens only in the liver, okay, where the glucose can be stored, right. In the other cells, what happens is, see, the enzymes glucokinase and glucose 6 phosphatase are both found in liver, but not in most other body cells. Why would it not be in most other body cells? Because then you wouldn't get the energy. Glucose has to be broken down. If all the steps prevented the breakdown of glucose, then obviously you would not get energy. But in the liver, the excess glucose is stored as glycogen, okay, because there are these specific enzymes that are present in the liver that can form glycogen from glucose. Okay. So, what we actually looked at is, we looked at the first few steps of glycolysis, where what we have ultimately come to today is we have broken down the 6-membered ring of glucose to 2 3-membered rings. Now, we are going to see how those 3-membered rings, or rather glyceraldehyde 3-phosphate, actually will form pyruvate, okay, and finally, how that in anaerobic conditions goes to lactate or goes through acetyl-CoA to the tricarboxylic acid cycle, where it is finally broken down. We will see that in our next class. Thank you. Continue our lecture on glycolysis. We started off yesterday where we considered the different metabolic processes that actually go on in the body, and if we look at the first slide here, the overall catabolic processes look at the breakdown of lipids, polysaccharides and proteins. Now, the breakdown of lipids gets into components of fatty acids and glycerol. The polysaccharides break down to monosaccharides and the proteins break down to amino acids. Now, in the anabolic processes, we have these broken down amino acids and other factors that actually get on into building up the other macromolecules in the that are required for our bodily functions. Now, what we are interested in is the breakdown of the monosaccharides, particularly the process of glycolysis that takes glucose and breaks it down into pyruvate, and later on we will see how this pyruvate then gets on into the tricarboxylic acid cycle or the Krebs cycle to finally get to water and carbon dioxide. And an offshoot of that is the production of ATP, where we studied oxidative phosphorylation in the different complex processes that require a number of electron transfer cofactors as well as certain enzymes. The overall equation for glycolysis is the breakdown of glucose into 2 pyruvates. Now, what we have here is we see how ATP is produced. We will see as we continue with all the steps, we will see how ATP is produced in some of the steps, but in the first set that we did, we found ATP consumption. This is the number of steps that we considered in our last class. We had glucose going to glucose 6-phosphate, the enzyme being hexokinase. We have to remember that a kinase is a transferase that transfers a phosphate group. So, in this process we had the breakdown of ATP to ADP and the phosphate was transferred to the glucose. This then went on to form the ketose from aldose. So, we had fructose 6 phosphate that had the enzyme phosphoglucose isomerase acting on it, because this is an isomer, the aldose and the ketose that we have here. The fructose 6 phosphate then went on to form fructose 1,6 bisphosphate, where we required another ATP to be broken down, and the enzyme used there was another kinase, but in this time it was phosphofructokinase. After this particular step, we have aldolase come into the picture. Aldolase is what actually breaks up the 6 carbon membered ring into 2 3 carbon membered rings. Getting oxidized in this case, glyceraldehyde is getting oxidized to form glycerate. What is getting reduced? NAD plus is getting reduced to NADH, and the enzyme is the dehydrogenase. Okay. So, we have glyceraldehyde 3 phosphate going to glyceraldehyde 3 phosphate dehydrogenase, and in the event it forms 1,3 bisphosphoglycerate. Now that you have formed the glycerate, you have to lose the phosphate to form the pyruvate. Okay. So, the first step in the loss of one of the phosphates is a phosphoglycerate kinase that is going to lose the phosphate that is attached to the carboxylic first carbon atom of the glycerate. Okay. And you have 3 phosphoglycerate formed. Fine. After you form 3 phosphoglycerate, there is a mutase reaction which shifts the phosphate moiety from the 3rd carbon to the 2nd carbon. So, you have 3 phosphoglycerate form 2 phosphoglycerate, then another enzyme that helps in the dehydration is enolase that results in phosphoenol pyruvate. So, after phosphoenol pyruvate, you have the enolic form of pyruvic acid, which then forms pyruvate after the loss of the phosphate. And who takes up this phosphate? ADP. Okay. So, that comprises the whole series of steps where glucose is broken down into pyruvate. Okay. Now, what we have to see is we have to do a balance of energy. Okay. We have to see how many ATPs are taken up, how many ATPs are produced, and to see whether the actual breakdown of glucose is giving us any energy at all. Okay. There is one thing that we have to remember, that per glucose there are 2 glyceraldehyde 3 phosphates. Okay. Because what is the previous step? We have 2 of these. Okay. And in the triose phosphate isomerase, we know that the equilibrium is shifted to this side because this is being consumed in the further steps.